Hello there, I'm Anika. So today we'll be discussing on the topic immunoglobulins. First we look into the definition of an immunoglobulin and then we look into the mnemonic to classify this immunoglobulin, which is also called immunoglobulin subtypes or antibody isotypes. And then we look into each of these subtypes based on their function, characteristics, half-life, etc. So we'll be going through each of these subtypes based on the order of the mnemonic. So let's start. Immunoglobulin. So it's made up of two words, immuno and globulin. Immuno means it has something to do with the immune system. And globulin means glycoprotein. It is a type of glycoprotein. So immunoglobulin together means a glycoprotein present in the immune system or a glycoprotein that functions in the immune system. The type of globulin present in immunoglobulin is alpha globulins. And they are synthesized, immunoglobulins are synthesized in the plasma cells. This is important. And the next important point is all antibodies, I have denoted it as AB. All antibodies are immunoglobulins. I have represented it as Ig. But all immunoglobulins may not be antibodies. So it only works one way. All antibodies are immunoglobulins, but all immunoglobulins may not be antibodies. So this was the first point. We looked into the definition, and now we're going to look into the structure. So the second point is structure. So an immunoglobulin molecule is made up of 4 peptide chains, and they have 2 light chains and 2 heavy chains. The light chains is identical in all immunoglobulins. It remains the same. It's the heavy chain that varies. Now both light chain and heavy chain is divided into two. That is constant and variable region. Constant and variable region. Both of these have a constant and variable region. And the light chains are of two types. The first one is kappa, represented as K. The second one is lambda. And also the other important point is the light chains and the heavy chains are linked by disulfide bonds. So this is how you represent it. Okay, so we have finished definition, and the second one is finished. We have finished the structure. Now we are going to look into the classification. So 3, classification. So immunoglobulins are classified based on their structure. This is very important. Immunoglobulins are classified based on their structure and also antigenic nature. I have written it as Ag, okay? Ag means antigen. Antigenic nature of the H chain. So it's classified based on the structure and antigenic nature of the H chain. So the classification, before that I'll tell you about the mnemonic used here for the classification. It is gamed. G stands for immunoglobulin G, A stands for immunoglobulin A, M stands for immunoglobulin M, E stands for immunoglobulin E, and D stands for immunoglobulin D. So this is very important. Remember the mnemonic game. There are 5 antibody isotypes of immunoglobulin, that is IgG, IgA, IgM, IgE, IgD. So we'll be discussing based on this order from IgG to IgD. So we have completed the first 3 important points, that is the definition, the structure and the classification. Now we're going to look into the subtypes. So the first subtype is IgG. And IgG is the most abundant. Most abundant where? So it's the most abundant in the serum. And 80% of the total immunoglobulins. It forms 80% of the total immunoglobulins. So where is it found? It's found in the blood, it's found in the plasma, is also found in tissue fluids. Now about the half-life. Half-life of IgG is 23 days. This is very important. This is the longest. None of the other immunoglobulins last for this long. So the half-life of IgG is the longest and it's 23 days. The IgG crosses the placenta. This is very important. It crosses the placenta and provide natural immunity. So IgG crosses the placenta and provides natural immunity to fetus, and also not just to the fetus, it also provides natural immunity to neonates at the time of birth. IgG acts against bacteria and viruses also, not just bacteria, viruses. By which process? By the process called opsonization. It also neutralizes toxins. There are 4 types of immunoglobulin G. That is type 1. We are not going to look deeply into it. I am just saying the subtypes so that you know about it. IgG1, IgG2, IgG3 and IgG4. These are the subtypes. Now it also, another important point, it activates complement. It activates complement by classical pathway. We know there are 2 pathways, that's the classical pathway and the alternate pathway. So the IgG activates complement by classical pathway. Now we're going to look into IgA, the second part of the mnemonic. So over here we said 80% of immunoglobulins is IgG. 10 to 15% of immunoglobulins is IgA. Where all is it present? It's present in the milk, it's present in the saliva, it's present in the tears, it's present in the mucus of respiratory tract, it's present in the digestive system, digestive tract, and also in the genitourinary tract. So it's present in the milk, saliva, tears, mucus of respiratory tract, digestive tract, and also in the genitourinary tract. The half-life of IgA is 6 to 8 days. So before it was 23 days for IgG, over here it's 6 to 8 days. The functions, now about the functions. The functions of IgG include, it provides local immunity. The second point is secretory IgA. So basically secretory IgA has 3 functions. The first function is secretory IgA. There are 2 types of IgA. The first one is the monomer and the second one is the dimer. This dimer is called secretory IgA. So secretory IgA binds to surface antigens. I have represented antigens as Ag. microbes and prevents its attachment and invasion of the mucosal surface of both respiratory and gastrointestinal tract or digestive tract. This is called immune elimination. The second function is secretory IgA provides an important, very important line of defense against a few microbes like Salmonella, Vibrio cholera, then influenza virus, and also poliovirus. The third function of secretory IgA is that it is present in the secretory IgA is present in the breast milk. And what does this do? And therefore protects newborn. Therefore protects the newborn during first few months of life. That's a very important function. It's present in the breast milk and therefore provides the newborn enough antibodies, protects the newborn, therefore protects the newborn during the first few months of life. The next function is about complement system. It activates complement by alternate pathway. Before it was classical for IgG, for IgA it is alternate. The next important point is promotes phagocytosis and also intracellular killing of microbes. The third one is IgM. So 5 to 10% of total serum proteins is made up of IgM, consists of IgM. The half-life is 5 days. 80%, this is different, 80% of the IgM is present intravascularly. And this IgM, this cannot cross placenta. So if there's like presence of IgM, in the newborn, I've written it short, newborn presence of IgM. That means if the antibody IgM is present in the serum of the newborn, it indicates that the newborn has a congenital infection. The next point is, it is the earliest immunoglobulin to be synthesized by the fetus. This is also very important. It is also the first immunoglobulin to be produced in primary response. So it's like the first immunoglobulin to be produced in primary response of antigen. If an infection occurs to the baby, or if an infection takes place, if there's an invasion by an antigen, the primary response is done by IgG first, and by IgM first, and then by immunoglobulin G. The next point is that IgM is short-lived. As I said, the half-life of IgM is 5 days, and since it's short-lived, if it's still present in the serum, so serum presence of IgM indicates infection. The functions of IgM include: it agglutinates bacteria, it activates complement by classical pathway, just like IgG. IgM does it by classical pathway, IgG does it by classical pathway, and IgA does it by the alternate pathway. It causes opsonization and also hemolysis, immune hemolysis for IgE. So IgE, the structure of IgE is exactly the same as IgG, or it's more or less the same, similar to IgG. It has a half-life of 2 days. The normal serum concentration of IgE is 0.3 microgram per ml. It does not cross the placenta. It is produced, where is IgE produced? It is produced in the lining of respiratory tract. Also, it is produced in the lining of the respiratory tract and also the intestinal tract. The next important point is the IgE is also called reagent. It is called reagent antibody. IgE does not does not activate complement, nor does it agglutinate antigens. IgE mediates immediate hypersensitivity, reaction. IgE is responsible for symptoms of anaphylactic shock fever and also asthma. So symptoms of anaphylactic shock, hay fever and also asthma. It plays a major role in immunity against helminths or helminthic parasites. IgE also releases histamines. This is what all of us know. It releases histamines, helps in inflammation, or it is an inflammatory mediator. The fifth one and the very last one is IgD. So IgD, exactly like IgE, the structure of IgD is similar to that of IgG. The serum concentration of IgD is 30 microgram per milliliter. 0.2% of the total immunoglobulins present is IgD. And the half-life of IgD is 3 days. So we have completed the topic immunoglobulins. So that's it. Thank you and have a nice day.